Hello and welcome. This is an introduction to experiment one. This experiment deals with studying the kinetics of an oxidation reaction of alcohols with dichromate ion. So let's take a quick scroll through the experiment so we see a brief introduction to some of the important equations about kinetics that we're working with in this experiment. The key reaction that we're looking at is dichromate reacting an acid in the presence of alcohols to produce ethers or ketone compounds through the oxidation reaction of the alcohol group, and then forming free chromium ion and water. So you can see in this reaction, dichromate's consumed, acid is consumed, the alcohol is consumed, and we produce the, in this case, aldehyde, free chromium ion. We only have really one type of color change taking place from the chromium, that's yellowish. Acid and alcohols are obviously colorless. And then we're producing colorless compounds except for the chromium ion, which is green. So we can see through following this reaction with UV vis, the change of the absorbance from something yellow-orange to more greenish, maybe slightly purplish as the reaction takes place. Ideally, we're going to find the rate law of this reaction, so we're going to try to find the order of the reaction with respect to dichromate. This is what we follow the reactions with, so we're going to keep this concentration relatively low. We're going to keep the acid and alcohol concentrations much higher, even though their concentrations can change during the course of the reaction. These concentrations will be so high that they can't effectively change. We'll discuss this as like pseudokinetics. So the substance that we're studying, we're going to first determine its order through looking at its color changes, and then from there try to observe the difference of the rates of the reaction when we change the concentrations of acid and the alcohol. We'll also look at different types of alcohols in this experiment. We'll look at the kinetics of the reactions of methanol, ethanol, 1-propanol, and isopropanol. One quick note would be that we're going to provide you with the solutions that you need to use. So instead of you guys preparing your solutions, we're going to do that for you. So you'll come into lab and have these solutions already prepared. If you have to describe in a narrative how you prepared the solutions, just say you use the solutions as they were provided to you. You might note that the specific concentrations aren't exactly the 0.0200. This will probably be a little bit off, but just use the concentrations as they're provided to you on the bottles that you're using in the laboratory. The stop flow apparatus has some separate videos on how to actually carry out some of these experiments. Just for a quick introduction here, a stop flow apparatus, the idea here is that you can flow two reactants together and then stop their flow and then monitor what's going on. So that's, in a nutshell, the stop flow kinetics apparatus. You can manually flow the liquids together. We'll show you that in a video, or you can use the computer and a set of pneumatics to actually do that for you to mix at some period of time. In one of the cases, we're going to manually add the reactants together and then try to observe the reactions that takes place. Let me actually step back real quick and say that what we're going to take a look at first is data. We're going to collect some data that allows us to do and produce a Beer's law plot of how the concentration of dichromate, what the absorbance of the dichromate is as a function of the concentration of dichromate. One of the things that you're going to do is you're going to have the dichromate in one syringe on the apparatus and then acid in the other. So the dichromate and the acid are actually going to be diluting each other. So be sure to account for that dilution here, that if you have 0.02 molar dichromate in one of the syringes and it's being flowed and mixed with another syringe containing the sulfuric acid solution, that these two concentrations would actually be 0.01 molar and then 1 molar for the acid. So be sure to actually consider the true concentration of dichromate when we're observing it on the apparatus. Continuing with the experiment, we'll actually look at the reaction on the apparatus itself. We'll mix together ethanol and acid. Now we actually need ethanol to react with the dichromate, and we also need acid. We've looked at this in the past and played around with this. We don't really have a whole lot of time to waste this semester, but if you just have alcohol and the dichromate, they effectively don't react together. You need to have the acid present. And we could actually put the acid with the dichromate or with the ethanol or with both. This semester, what we're going to do is mix the ethanol and acid together into one syringe. Then in a second syringe, we're going to have the dichromate. And then we're going to flow them together, and we're going to keep that mixture in the apparatus. And then every 4 minutes, we're going to take a look at what's going on during the course of the reaction. So we're going to get a sense in about 20 minutes of what peaks are disappearing, what peaks are growing in, just kind of verify that we're really seeing the loss of the yellow-orange color and the formation of some new color that we can observe at a different wavelength in the UV-Vis spectrum. Another thing we can do is actually use kinetics. We can follow the reaction at 440 nanometers. That's one of the sort of peaks that dichromate strongs at relatively, or one of the peaks that dichromate absorbs at relatively strongly. There's actually a more intense peak at about 350. We think that that peak's a little too intense to follow relatively high concentrations. I think what you're going to find through your reaction observation is that these reactions aren't incredibly fast. So we want to try to keep the concentrations high so that the reactions can be as fast as possible. We mentioned doing 25-minute trials. We're just going to do 10-minute trials because you're going to be working by yourself. We're going to have to get 2 students probably through this experiment depending on how big our class is. But we're going to have probably one of you go first in the day, the other one of you go second. We'll work that out as the experiment goes along. But do 10-minute trials here instead of 25-minute trials where we're taking the acid ethanol and the dichromate. So if you notice from the reaction observation and the reaction kinetics, we haven't changed anything. So we're using the same syringes so far. We're going to do a 10-minute trial, fit the data to a single exponential, and then we're going to repeat with a lower concentration of dichromate, keeping the same ethanol and acid syringe loaded up. So we're going to get right off the bat here the data that shows us how fast this 0.02 molar reactor, remember it's really 0.01 once it dilutes with the other syringe. Then we're going to take half the concentration of dichromate, react it with the same acid alcohol mixture, and sort of compare those rate constants. So if the reaction is slowing down, we should see a lower rate constant. And the question is, does it lower by about a factor of 2, or by a factor of 4, by a factor of 16? Is the order with respect to dichromate, is it going to be 1, is it going to be 2, is it going to be 3? You should get that data pretty clearly right off the bat in this experiment. And then from there, you can export your data. So export the data from your kinetic runs and also from your absorbance scans. Prepare Beer's law plots, and you can do most of this work here at home or over in the computer lab. The adding water to the syringes, this is just like rinsing some water through once you finish with your experiment. It's a good idea to leave water in the lines of the apparatus as opposed to high concentrations of ethanol. So at the end of the first day, what you're going to be able to do is you're going to be able to write a narrative based on some of your results. Again, you don't have to describe how you made the solutions because you're going to use them as prepared. And then you can explain, though, how you use the stop flow apparatus and then show and discuss your plots and your results from that first day. Okay, and so a narrative is, we're going to talk about this in class, but a narrative is something that explains to us what you did. So it should be, in this experiment, I mixed together these chemicals for this amount of time, did this to them, and I observed this, and from that I can conclude this. So it's a summary of what you had done and accomplished here in this first day. As we move into day 2, you're going to look at the reactions as you vary the concentrations of the ethanol and the acid. So you get a sense of how does the reaction change if you change the concentrations of acid and ethanol, and what if you use different types of alcohols. So here's our comparison of the different reactions with different types of alcohols. And then from there, you're pretty much just doing some data analysis and finishing up. So there's really just 5 new trials that we do on day 2. 2 of the trials are already done on day 1. You can just feed those results in. And then from there, you're just doing some analysis. And then day 3, you can wrap up anything that didn't go well. So from, I feel like there's a page missing. I don't know where page 8 went, but there's a page 8 somewhere. There it is. For day 3 is you're just repeating anything that was maybe ambiguous, and then you're preparing your final presentation and conclusion from there. Okay, so that's a quick introduction to experiment 1. Hopefully this gives you a good start or a good ability to come in the lab and get started on the first day of the experiment.